Hello everyone, my name is Uzair Hashmi and for my profile details please visit sqadia.com. So let's get started about this lecture. In today's lecture we are going to discuss about the renal regulation of all of these ions including calcium, potassium, phosphate and magnesium ions. Along with their renal regulation, we are going to discuss its integration and mechanism of regulation of how these ions play their important functions in the maintenance of the extracellular fluid volume and the pressure. So in this lecture, we are going to discuss first of all the renal excretion and regulation of potassium in the extracellular fluid. We are going to discuss how what is the normal quantity that is present inside the cell and outside the cell in the extracellular fluid. We are going to discuss the regulation of internal potassium distribution. That is, whenever you intake potassium, let's say for example, the extracellular volume, the amount of potassium in the extracellular fluid is dangerous, it's lethal, so the cell has to take the excessive potassium inside the cell. And then we'll have an overview of renal potassium excretion, how after it's being regulated, under what circumstances the excretion of renal potassium would be increased and under what circumstances it would be decreased. After that, we are going to discuss the potassium secretion by the principal cells of late distal and the cortical collecting tubules. We are going to discuss under what circumstances, after they are being reabsorbed from the proximal and the distal tubules, what are the mechanisms that are involved in the secretion of the potassium in both from the late distals and the cortical collecting tubules. And then we are going to discuss the summary. We are going to summarize what are the factors that play their part in the regulation of potassium secretion. And then we're going to discuss the extracellular fluid potassium concentration, and we're going to discuss how when a particular amount of potassium reaches a threshold, what happens or what changes occurs in the body that regulate or that stimulate the secretion of potassium in the urine. After that, we're going to discuss the control of renal calcium excretion, like we discussed the potassium ion secretion and regulation. We're going to follow after calcium the control of regulation of magnesium ions and the extracellular magnesium ion concentrations. After that, we are going to discuss the pressure natriuresis and pressure diuresis. We are going to discuss what happens when the amount of potassium, what happens when the amount of sodium is abundant, and under what circumstances the amount of sodium would increase in the urine, in the tubular fluid, which of course ultimately results in the excessive amount of water in the urine, resulting in the formation of dilute urine, because the sodium increases the osmolarity of the urine or the tubular fluid, which is going to be converted into urine. So this of course will cause the movement of the water or retention of the water within the tubular lumen and not reabsorb back. So at that particular scenario, even the antidiuretic hormone would not be secreted. After that, we are going to discuss the renal body fluid mechanism, and finally we are going to discuss the role of the renin-angiotensin system. So in this slide, we are going to discuss the regulation and excretion of potassium in the extracellular fluid. So let's get started. In this particular figure, you can see that the extracellular fluid contains about 4.2 milliequivalent of potassium as compared to the inside, where inside contains about 140. So the concentration of potassium in the extracellular fluid is 4.2 milliequivalent per liter. So many of the cell functions, they are very sensitive to even minute changes in the potassium concentration. a small increase or decrease in the potassium concentration inside the cells, they can trigger many abnormalities. For example, if you have an increased concentration of potassium in the plasma or in the blood, and that increase is only from 3 to 4 milliequivalents. Let's say the normal concentration is 4.2, it increases to 7.2 or even 8.2 or even up to 9. It could cause the cardiac arrhythmias, and even higher concentration would be much more disturbing for the body. For example, in this particular case, the case of heart, it can lead to cardiac arrest, myocardial infarction, or the fibrillation. Now let's take a case scenario where you have a 70 kg adult that has about around 28 liters of the intracellular fluid. Now this 28% of the intracellular fluid accounts for the 40% of its total body weight. So out of these, for 28 liters of the intracellular fluid, we have 14. It's against we have 14 liters of the extracellular fluid, and now that accounts for the 20% of the body weight. Now about 3920 milliequivalent of the potassium are inside the cell. 3290. That is much more as compared to the amount of potassium outside, and only in the extracellular fluid about 59 milliequivalent of the potassium concentration is present in the extracellular fluid. And along with that, we have a daily intake of about ranges from as low as 50 to as high as 20 milliequivalent per day. So here, if we intake 100 milliequivalent of the potassium ions per day, it will move to the extracellular fluid, it will move down to the intracellular fluid, so that the there is no immediate rise in the plasma concentration of the potassium. So after a particular when the day has been passed, potassium output in the urine would be about 92 milliequivalents, and in the feces is much low, and that is about 8 milliequivalent per day. So the intake equals the outtake or the output. So how does the internal potassium distribution is being regulated? In this slide, we are going to discuss that after you have ingested the potassium through various variety of foods or the fruits, the extracellular fluid potassium concentration would of course rise to a level that is lethal to a body. So something has to be done in order to prevent the body from the lethal rise of the concentration of the potassium. So it has to be taken inside in the cell whenever you have intaken or ingested enough or high levels of the potassium. So there are several factors which will regulate, of course, the cellular uptake of potassium after it's being ingested. Number one is the insulin. Now, insulin plays a very important role in the potassium uptake of cell, because what happens is that whenever you can have a very high increase in the potassium concentration, insulin would be activated along as by the pancreas. So it will, of course, along with the regulation of glucose, it would also cause the regulation or the upregulation of the uptake of the potassium by the cells. So what happens in case of the diabetes mellitus, because there would be no enough levels of the insulin, so along with the increased concentration of glucose, there would be increased concentration of potassium as well, and that would lead to a condition or complexity known as hyperkalemia, which of course means that there is high concentration than normal concentration of potassium ions in the extracellular fluid. Now, the aldosterone. Aldosterone increases the potassium uptake of cells. Aldosterone plays an important role in the regulation of the sodium in the water and the chloride in the water. But along with this regulation of the sodium, it also plays an important role in the regulation of potassiums in the cells. So there would be, of course, increased potassium intake when there would be the aldosterone secretion, which will of course cause the increased cellular uptake of potassium. Now, the beta-adrenergic stimulation plays a very important role as well in the cellular uptake of potassium. Now, this happens when there is the increased secretion of the catecholamines, and one of the catecholamines is the epinephrine, or the non-epinephrine as well. In this particular scenario, when the epinephrine is being secreted, it would cause the movement of the potassium ions from the extracellular fluid into the intracellular fluid. And this will occur, of course, by the activation of the beta-2 adrenergic receptors, which will, of course, cause the simulation or the inward movement by creating a concentration gradient or the electrochemical gradient by the movement, which would, of course, result in the movement of potassium ions from the extracellular fluid into the intracellular fluid or within the cells. Now, one of the important factors is that whenever you will have an acid-base abnormalities, these abnormalities of the acid base, either in the case of alkalosis or the metabolic acidosis, you will have, of course, there would be changes in the concentration of the potassium in the extracellular or the intracellular fluid. So what happens in the metabolic acidosis case, of course, metabolic acidosis is a case when there would be increased concentrations of the acids. And that means that because acid is a species that donates proton, so basically there would be increased concentration of protons. Now, in case of the metabolic acidosis, there would be, of course, increased in the extracellular potassium concentration. Now, increased in the extracellular potassium concentration, if you would recall, I told you earlier that increase in the extracellular potassium concentration can be lethal, so something has to be done to avoid this particular lethal increase. But what happens in case of the metabolic acidosis is that the concentration of potassium that is required in the extracellular fluid, that is about 4.2 milliequerels per liter, that decreases to the normal concentration. So in case of the metabolic acidosis, the extracellular potassium concentration would, of course, decrease. Now, what happens in case of increased hydrogen ions? In case of increased hydrogen ions, that is the case of the metabolic acidosis, this would cause the decrease in the activity of sodium potassium ATPase activity. So if you would recall, sodium potassium ATPase, what it does is it causes the outward movement of the sodium from inside to the out of the cell, and similarly 2 molecules of potassium moves inwards as a result of the ATPase activities of the sodium potassium pump. So what are the factors that would regulate the internal potassium distribution? It's being continued. Let's start with the cell lysis. What happens in the case of cell lysis, there would be increase in the extracellular potassium. Now, cell lysis can occur due to a variety of reasons. It can occur due to cell lysis can occur as a result of the necrosis or any inflammation that would have activated the complement system or the innate immunity, which would of course cause the phagocytosis, release of the cytokines, pro-inflammatory cytokines. These cytokines will of course cause the cell lysis and the inflammation in that particular microenvironment. Now this would of course cause the cell lysis. Now this cell lysis would of course take out all the constituents that are present within the cell outside the cell. So the amount of that is around 140 milliequerels per liter of the day. The concentration of potassium which was inside the cell would be of course released outside the cell as a result of cell lysis, which would of course can lead to the potential lethal rise in the potassium levels. So of course, it's been written over here as well that the large amount of potassium contained in the cells are released as a result of cell lysis into the extracellular compartment. The second one is the strenuous exercise can cause the hyperkalemia by releasing the potassium from the skeletal muscles. Now, what happens in the case of strenuous exercise is that there would be, of course, strong contraction of the muscles, and strong contraction of the muscle requires enormous increase in the activity of the sodium-potassium pump, because the sodium inward movement of the sodium is required for the release of the calcium, so that whenever the calcium will be released, it would be binding to the troponin head, as a result of which muscle contraction would occur. So whenever the sodium will be moving inside, it would cause the outward movement of the potassium as well. So this outward movement of the potassium will of course increase the levels of the potassium in the extracellular fluid. So what happens in that is that the strenuous exercise, or this particular kind of exercise, which would of course burn lots of calories, would cause increase in the potassium concentration in the muscle extracellular fluid. Now, the other reason is that the increased extracellular fluid osmolarity. This plays a very important role in the redistribution of potassiums from the cells to the extracellular fluid. Now, what happens is that if we have increased potassium concentration outside the cell as compared to inside the cell, so there would be much more osmolarity, or the solute potential outside the cell as compared to inside the cell. So as a result of osmosis, the water will be moving from inside to the outside the cells as a result of increased osmolarity of potassium ion concentrations. So let's have an overview of how the renal potassium excretion is being regulated and maintained. Now, there are three important steps that would be determining the process of the renal excretion. The first step is the rate of potassium filtration by the glomerular filtration rate, that is multiplied by the plasma potassium concentration. As there would be increase in the rate of potassium filtration, there would be increase in the renal secretions as well. The second includes the rate of potassium reabsorptions by the tubules. I'm going to discuss this particular figure after I have discussed the third point as well. The third point includes the rate of potassium secretion by the tubules. So the first step is how much quantity of the glomerular filtrate contains the potassium. So the greater the concentration of plasma, the greater the concentration of potassium in the plasma, the greater would be the filtration rate, of course. So greater amount of potassium will be moving inside the glomerulus through into the tubules. Now, of course, the second case would be involving the rate of potassium reabsorption. So the potassium is reabsorbed from the proximal tubules and the ascending tip portion of the loop of Henle. These two points cause the rate of determine the basically rate of reabsorptions of the potassium from the proximal portion and the ascending tip portions of the loop of Henle. Number third is the late distal portion of the proximal tubules, late distal portion of the tubules. They would of course cause the secretion of the potassium under circumstances where the extracellular fluid has enough quantity of the potassium. So over here, if we have 756 milliequivalent of the potassium concentration, 65% would be reabsorbed from this particular portion, the 27% would be reabsorbed from this particular portion, and 4% of the potassium will be secreted from the distal portion, about 4%. So this would ultimately cause the 12% of the potassium would be excreted out to regulate the normal concentration of potassium inside the cell and outside the cell as well. So this particular diagram explains the potassium secretion by the principal cells, and these principal cells are the cells that are present in the late distals and the cortical collecting tubules, as I have discussed over here. This arrow explains the principal cells that are present in the late distal tubules, and this particular arrow will be representing the potassium ion excretion by the cortical tubules. So what we have over here is the sodium potassium pump. This would be causing the inward movement of the potassium as the outward movement of the sodium is going to take place. So this would cause the increase in the potassium ion concentration inside. So this would cause, of course, this would produce an electrochemical gradient where potassium has high ion concentration inside as compared to the outside. So what would be happening is that the potassium ions would be leaking outside as a process of diffusion through these channels into the tubular lumen. So these would be happening from the renal interstitial fluid into the principal cells, and these principal cells of the late distals and the cortical tubules would be secreting potassium into the tubular membranes or the tubular lumen. So to summarize, the potassium secretion is controlled by the principal cells of the late distals and the cortical tubules. It is regulated by the activity of the sodium potassium ATPase pump, because that is taking the potassium inside the cell when it is taking the sodium outside. Of course, the second important step that I discuss is the establishment of the electrochemical gradient from the blood to the tubular lumen, from the blood towards the tubular lumen. And of course, the important third point, which cannot be minimized, is the permeability of the lumen membrane for the potassium. It cannot be negated and it cannot be overlooked. This particular point plays a very important role, because the permeability of lumen for the potassium is much more than its permeability for sodium. So a large amount of sodium can be taken outside into the tubular lumen as compared to the sodium. However, the sodium reabsorption from the tubular lumen takes place when the potassium is being secreted out into the tubular lumen, which will of course be going into the urine. So let's have an overlook on the summary of the factors that regulate the potassium secretion. Now we have discussed all of these, so I'm going to summarize in this particular slide the factors that of course regulate the potassium ion concentration. These include, number one is the increased extracellular fluid potassium concentration. When you have increased extracellular fluid concentration, this plays a very important factor, because it would cause reduction in the sodium absorption, reabsorption, and increase in the sodium secretion. So to maintain the normal physiologic concentrations of the potassium in the cells and in the extracellular fluid. Increased aldosterone plays a very important role when it causes the reabsorption of the sodium from the late distal and the cortical tubules and the water reabsorption. It simultaneously increases the tubular secretion of the potassium. And of course, increased tubular flow rate plays a very important function as well. One important factor that would decrease the potassium secretion is the increased hydrogen ion concentration. That is the case of acidosis. So what happens in case of the acidosis is that there would be, of course, increased hydrogen ion concentration. This increased hydrogen ion concentration would ultimately cause the increased deactivity or the deregulation in the presence of the hydrogen ion. What would happen is that increased hydrogen ion concentration would be causing the decreased activity of the sodium potassium ATPase pump. So what would happen in that particular case is that not much sodium would be taken inside the cells, so they won't be secreted out. So this would of course cause the decreased potassium secretion. So what are the factors that would be stimulating the potassium secretion? Now let's read through these points one by one, and then we're going to discuss each point. So it says that increased extracellular fluid potassium concentration stimulates the sodium potassium ATPase pump. So when the sodium potassium ATPase pump would be increased, its activity would be increased, there would be increased uptake across the basolateral membrane. Now this would in turn of course cause the increased intracellular potassium ion concentration and which would of course cause the diffusion across the luminal membrane into tubule. So this point explains that when there is increased extracellular fluid concentration of potassium, when the potassium is much more in the abundant quantities in the extracellular fluid, it would be activating the sodium potassium pump. So now when the sodium potassium pump is activated, it would be causing the inward movement of potassium inside the cell. So when the potassium has moved inside the cell, it would cause the enough increase in the intracellular concentration of potassium as compared to outside the basolateral membrane. So this would establish the electrochemical gradient. Now this increase in the electrochemical gradient will cause the diffusion or leakage of the potassium ions from inside to the outside of the cells into the tubular lumen. So there would be, of course, increased diffusion from inside the cell into the tubular lumen through the basolateral membrane. So there would be, of course, increased concentration of potassium in the tubular fluid, which would be depicted in the high concentrations of potassium in the urine. So point 2 says that increased extracellular potassium concentration would increase the potassium gradient, of course, that I have discussed. When there would be increased extracellular potassium concentration outside as compared to inside, so this would create an electrochemical gradient. This concentration gradient would cause the movement of potassium into the cells. Of course, this would reduce the breakage of the leakage of the potassium ions from inside the cells through the basolateral membrane. Now this part plays a very important function, that the very first step is the movement of potassium from outside the extracellular fluid into the basolateral membrane and into the tubular cells, epithelial cells. Now in this particular case, because the concentration over here, if I can show you through the diagram, let's say in the renal interstitial fluid, the concentration of potassium has increased to up to a particular level. So it would cause the activation of this, and because the electrochemical gradient has been established, the potassium will be moving outside. At that particular situation, when the concentration of potassium is high over here as compared to over here, this would, of course, block the leakage of the potassium to move from over here to over here at that particular case, or even the active transport back to the renal interstitial fluid. So there would come a time when the concentration of potassium inside the principal cell would, of course, increase as compared to the concentration in the renal interstitial fluid or the extracellular fluid as compared to the principal cells. So at that particular case, the function of this sodium potassium ATP pump would be completed. Now the next step would be establishment of the concentration gradient from the potassium over into the cells to the tubular lumen, because the tubular lumen has much less quantity of potassium as compared to the potassium inside. So this would again establish the concentration gradient, but now this concentration gradient would be established between the principal cells and the tubular lumen. This increase in the tubular lumen concentration of the potassium would ultimately be caused by the increased in the movement of potassium from the principal cells into the tubular lumen. So this increase in the potassium concentration, of course, will be sending signal to produce much more aldosterone, and this aldosterone will be, of course, secreted by the adrenal cortex, and this would again increase the permeability of the cells to secrete potassium, which would, of course, further stimulates the potassium secretion from the late distals and the cortical portions. Now, the aldosterone, in the case of aldosterone, in this particular figure, first of all, let's have a look at the schematic diagram. So what happens when the potassium intake is increased, it would cause the increase in the plasma concentration of potassium. Now, it will do two things. The first thing, it will send signal to the adrenal cortex to secrete aldosterone. Now the aldosterone and the plasma concentration both simultaneously will cause increase in the potassium secretion by the cortical collecting tubules. When there would be increased secretion of the cortical collecting tubules, this would, of course, be causing the increased excretion or elimination of potassium ions into the urine. So over here you can see the effect of aldosterone. As the plasma concentration of aldosterone increases, the urinary potassium excretion increases in this particular diagram as shown in the red line. But in case of the extracellular concentration of the potassium concentration, the extracellular potassium concentration when increased over here, as you can see, the potassium concentration is increasing at that particular case when it would be increased up to 4 times of the 4 milliequivalents, because 4.2 is the normal concentration. When the concentration increases above beyond 4, because 4.2 milliequivalents per liter was the normal concentration of the potassium in the plasma. So when it was increased beyond that particular point, even before 5, between 4 to 5 times, what happens is that, because of course the plasma concentration of the potassium is reaching to a lethal limit, so the body needs to get rid of the excessive potassium before it reaches to that lethal concentration. So at that particular lethal concentration, it would be of course damaging to the body. So the body immediately acts by excreting out or increasing the excretion of the potassium concentration, as shown by a rapid increase in the potassium secretion or excretion than the normal. So this figure again explains that in the presence of the normal concentration, this would be the normal plasma concentration of the potassium. But what happens when the aldosterone mechanism system is blocked? When the aldosterone mechanism is blocked, the potassium intake, because the potassium intake is being increased, so as compared to this particular concentration, because there is no aldosterone, so the aldosterone won't be acting at the cortical tubules and sending signals that you have to secrete so potassium, because its concentration is increasing in the plasma, and that increase in the plasma potassium concentration is lethal. So this sends signals to the adrenal cortex then releases aldosterone then shrugs and activates the tubular portion of the cortical tubules, and cortical tubules then realizes that potassium needs to be secreted out. So in the absence of the aldosterone system, or the drugs that basically act as the antagonist of the aldosterone, the plasma potassium concentration can increase to up to a very lethal quantity. So in this particular diagram, we can see that the sodium intake, as the sodium intake has increased, it will do of course two three things. Number one, of course, it would decrease the aldosterone concentration, because aldosterone also functions for the sodium to reabsorb. So when the sodium concentration is increased, there would be less reabsorption of the sodium into the tubules. This of course will cause the increased glomerular filtration rate. Of course, that is due to the increased blood pressure as well. The proximal tubular portion of the sodium reabsorption will be decreased because sodium intake is increased, and the body needs to regulate the sodium to the normal concentration, to the normal range. Now both of these, increased glomerular filtration and decreased sodium reabsorption, would increase the distal tubular flow rate, because it has to be secreted out. So this would increase in the tubular flow rate. Now this increase in the tubular flow rate will of course promote the potassium secretion at one point, but decrease in the aldosterone concentration would decrease the potassium concentration. So both of these positive signals and the negative signals, there would be no net effect of the potassium secretion, because the positive stimulus that came for the potassium secretion was negated or minimized by the negative stimulus for the potassium secretion. So there would be no change in the potassium excretion whenever the sodium intake has been increased. So that is how kidney plays a very important function in regulating independently the solute concentration. That is, if the sodium concentration is increased, there is a mechanism that the potassium concentration stays the same, even when the sodium intake is coupled with the potassium outtake through the sodium potassium pump. So up till now, we have discussed the regulation of the potassium in the intracellular fluid and the extracellular fluid. We discussed how the regulation of the Potassium is being distributed internally within the cells. We discussed an overview of how the potassium will be secreted, and let's say it is being reabsorbed from the proximal and the ascending thick loop of Henle, and it is being secreted from the late distals and the cortical portions. We discussed the summary of the factors that basically regulates the potassium secretion. We also discussed in the last slide how the increased concentration of the potassium in the extracellular institution would of course stimulate the potassium secretion. Now we'll be discussing the calcium and magnesium concentration along with the pressure, natriuresis and the diuresis, and the renal body fluid mechanisms along with the renin-angiotensin system. So what happens is, in case of the calcium ion concentration, the calcium ion concentration, of course, along with the sodium, potassium, chloride, magnesium, phosphate concentrations needs to be regulated up to a particular range. So it does not exceed below the low range of the normal and beyond the above normal range, the upper limit. So what happens when there is low or less concentration of calcium? The condition arises known as the hypokalemia. This hypokalemia increases the excitability of the nerves and the muscle cells. Of course, this would of course cause the increased and extreme, resulting in the hypokalemic tetany or the contraction. This is characterized by the spastic skeletal muscle contraction, because of course the potassium ion the calcium ion concentration is low. Of course, this low concentration would increase the excitability, and when the excitability is increased, what would happen is that the increase in the excitability would send signals to muscles for the muscles to contract again and again and again and again. This would of course cause the muscle spasms, which can be lethal if that particular mechanism is occurring in the case of respiratory muscles. That can lead to choking. Now, what happens in case of the hyperkalemia? The hyperkalemia would of course depress the neuromuscular excitability. And now, whenever it would reduce the neuromuscular excitability, there would be no signal for the muscles to excite, so there would be no contraction of the muscles. That would lead to abnormal muscle contraction. While in case of the cardiac cells, it can cause the cardiac arrhythmias. So what happens? It can when the when the change of the plasma ion concentration, it can influence the degree of calcium binding to the plasma proteins. That is, whenever the pH reduces, because the hydrogen ion concentration is increasing, that means that the pH is going towards much more acidic. So the change in the pH, plasma pH, can change the binding of the plasma proteins with the calcium, because about 50% of the calcium binds to the plasma protein, and only left the remaining 50% is available for the glomerular filtration process. So what happens in case of the acidosis is that the less calcium is bound to the plasma proteins. So when less calcium is bound to the plasma protein, so this concentration of the free calcium would increase beyond the 50%. So there will be much more excretion of the calcium, which would of course cause the hypokalemia. Conversely, in case of the alkalosis, there would be great amount of the calcium that is bound to the plasma proteins, which is of course the opposite of the alkalosis. Therefore, what would happen is that in case of the patients with alkalosis, they are much more susceptible to the hypokalemic tetany, because enough concentration plasma protein contains more than normal concentration of the calcium. So this would of course reduce the concentration of free calciums in the plasma. So this would of course cause the hypokalemia, and that can lead to the hypokalemic tetany, which will be of course characterized by the muscle spasms. Now, whenever there is a case of low calcium concentration, this low calcium concentration would of course trigger the release of the parathyroid hormone. Now, this parathyroid hormones, it acts directly on the bones to increase the reabsorptions of the bone salts and increase in the secretion of the calcium. As you can see over here, The decreased stimulus of the calcium, the decreased calcium ion concentration sends signal for the release of the parathyroid hormone. Now it does three things. Number one is the activation of the vitamin D3. This vitamin D3 then ultimately increase the calcium reabsorption from the intestine, so that there would be much more increased concentration calcium reabsorptions in the intestine in the presence of high levels of the vitamin D3, which of course would be due to the increased parathyroid stimulation, and that would be of course due to the decreased calcium secretion. This parathyroid will of course cause at one time the intestinal calcium reabsorption. At the other hand, it would of course cause the reabsorption of the calcium from the renal tubules as well. Number three, of course, it would cause the release of the calcium ions from the bone, so that the body extracellular fluid can maintain its normal calcium ion concentration. So basically, the renal calcium excretion is basically the difference of the filtration of the calcium and the calcium which is being reabsorbed. So as I've discussed in the previous slides, that only 50% of the plasma calcium is ionized, which means that the remainder or the remaining calcium is bound to the plasma proteins, so they cannot be filtered. So only 50% will be filtered. Of these 50%, and the 99% of the calcium is being reabsorbed back by the tubules, and only 1% among that 99% of the 50% would be of course excreted out. Now let's have a breakdown of this 99% reabsorbed calcium. Now among these 99%, 65% is reabsorbed in the proximal tubule, 65% is filtered in the proximal tubule, reabsorbed in the proximal tubule, 25 to 30 is being absorbed from the loop of Henle, and major chunk from the ascending thick loop of Henle. And 4 to 9% is being reabsorbed in the distal and the collecting tubules. Now this pattern of the reabsorption is similar to that of sodium, because 65% reabsorption of the sodium takes place in the proximal tubule and 25% takes place in the ascending thick loop of Henle, and very low, about 4 to 10% of the potassium sodium ion concentration takes place from the distal and the collectical tubular portions of the body. Now, the phosphate plays a very important role in the regulation of the calcium, because calcium is basically present in the form of the calcium phosphate in the free ions forms, and the rest is being bound to the plasma proteins. So increase in the plasma phosphate concentration will of course stimulate the parathyroid hormone. This stimulation of the parathyroid hormone will be increasing the calcium ion concentration in the plasma by the increased reabsorption by the renal tubule, increased calcium absorption from the in small intestine, and by secreting calcium from the bones, to which will of course after the calcium ions are increased in the plasma, this would of course bind with the phosphate ions, which of course has been increased beyond the normal, so that calcium is present with the phosphate and not in the ionized form, which would of course can be damaging by the deposition of the calcium, forming kidney stones as well. Now let's have a brief overview of the factors that would either activate, upregulate or downregulate the calcium secretion. The parathyroid hormone, increased in the parathyroid hormone would be occurring when the calcium excretion is very low. Decreased extracellular volume in case of the extracellular secretion, where there would be low blood pressure, and when there would be high plasma phosphate concentration, excretion would be low of course, and that would be occurring in case of metabolic acidosis. In case of metabolic acidosis, the plasma hydrogen ion concentration increases, so calcium excretion will of course be decreased, and along with the vitamin D3. So opposite of what happens for these particular factors would of course increase the calcium secretion. So decreased parathyroid hormone, decreased quantity of the parathyroid hormone, increase in the extracellular fluid volume, increased blood pressure, decrease in the plasma phosphate, and metabolic alkalosis. All of these factors combined would be causing increased calcium excretion. Now let's have an overview of, we have discussed the regulation, excretion, and the concentrations of potassium ions. We discussed calcium ions, and now we're going to discuss about the magnesium ions. So the control of the magnesium concentration and the extracellular magnesium ion concentration. Now, more than one half of the body's magnesium is stored in the bones, of course. Although the plasma concentration of the magnesium is about 1.8 milliequivalents per liter, more than one half of this is bind to the plasma protein. Now, this binding of the plasma protein of the magnesium is similar to the binding of the binding of the calcium to the plasma proteins. So what happens is that therefore free ionized form is about only 0.8%, which is about half of the 1.8. That would be around 0.8 to 0.9 milliequivalents per day. So about half is bind to the plasma protein and the rest half is free. So that free amount of the magnesium is being regulated for excretion. If the quantity of magnesium increases or decreases, it's being regulated. So magnesium ion excretion would be regulated by, of course, changing the tubular reabsorption of the sodium or the tubular secretion of the sodium. This occurs, 25% of the reabsorption occurs in the proximal tubules. We have discussed for the calcium, this was 65%. 65% of the calcium was being reabsorbed in the proximal tubule, while in this particular case, the proximal tubule usually absorbs only 25% of the magnesium. Now, the primary site for the reabsorption of the magnesium is about in the loop of Henle, which accounts for the 65% of the sodium, while in case of the calcium, the loop of Henle was accounting for about 25 to 35%, and this percentage was same for the sodium and the potassium. So magnesium has about a slightly different mechanisms for its reabsorption preferences, where the proximal tubule reabsorbs only 25% and the loop of Henle absorbs a major chunk, that is about 65% is being reabsorbed. And these let's discuss in what particular situations there would be increased magnesium excretion. So number one is whenever there would be increase in the extracellular fluid magnesium concentration, this would reduce the reabsorption of the magnesium both from the proximal tubules and from the loop of Henle, and this would of course increase the sodium secretion to maintain or to regulate the normal concentration of the potassium of the magnesium. When there would be of course increased extracellular volume, this would of course be caused by when the extracellular fluid volume would be there because there is increased in the osmolarity, so that would cause of course the movement of the water in the fluid. This can also occur due to variety of reasons, including the hypertension or the edema. This can also occur as a result of diabetes insipidus, because the body would be producing a large quantity of the extracellular fluid. Of course, this would result in the excessive loss of the magnesium because the magnesium ions are not being intaken or ingested as compared to the fluid that is being ingested. So this would be resulting in the loss of magnesium by the production of excessive amount of urine, which of course we would be again stimulating much more loss until or unless something has to be done to keep or to conserve the magnesium ion concentration. The increased extracellular fluid calcium concentration of course results in the increased magnesium ions excretion as well. Now this let's discuss the pressure natriuresis and pressure diuresis. Now pressure diuresis refers to the effects of increased blood pressure and that would of course be due to the increased urinary volume excretion, would be leading towards the increased urinary excretion. Now what happens is that in case of the pressure natriuresis would result in the increased sodium excretion that would be occurring as a result of elevated blood pressure. So what is happening in that the increased sodium ion concentration would increase of course the blood pressure. It would also, so it would prevent the reabsorption of the sodium by the both the ascending loop of Henle and by the distal tubules and the cortical collecting tubules. So this, when the reabsorption is being prevented, the osmolarity of sodium would be increased. Now this increase in the sodium osmolarity would be causing the movement of water through osmosis from the principal cells or from the renal interstitium into the tubular lumen. That would of course cause the pressure diuresis as well. So let's have an overview at this particular figure. Over here you can see the effects of the atrial pressure on the sodium output. This is the pressure diuresis example. Over here you can see that the chronic increase in the atrial pressure. You can see the chronic increase in the atrial pressure, which would of course cause a much more greater sodium output as compared to the acute natriuresis. In this particular case, acute conditions can arise as a result of in this particular case, in case of the acute natriuresis, what happens is that there would be a simultaneous or the abrupt increase that can be taken aback, or that can be normalized. But in case of the chronic situations, the chronic conditions, because they are progressive, so they are very hardly or they are very mildly reversible. This of course causes much more damage as compared to the acute conditions as well. So let's have a look at the renal body fluid feedback mechanism. So what happens is that the peripheral resistance, when the peripheral resistance would increase, of course, okay, let's start from here. The fluid intake was increased and the non-renal fluid loss was increased, of course. This would, both of these would cause the rate of, this would of course cause the increased atrial pressure, and this would be atrial pressure would increase as a result of the total peripheral resistance when it will be increased. So the resistance would cause the increase in the atrial pressure. This increase in the atrial pressure would of course cause increased in the renal fluid excretion. This would of course cause the increase in the extracellular fluid volume, which would of course cause the increased blood volume. Now when the blood volume will be increased, so this would also cause increase in the mean circulatory filling pressure. This means that there would be much more increase in the volume of the blood in both atria and the ventricles. So there would be increase in the quantity of the blood volume, so it would of course cause the increase in the venous return, because the blood volume has increased, let's say from 6 to about 6.2 liters or 6.5 liters or up to 7 liters. This would of course, as the volume has been increased, and this would increase the venous return. So that is whenever the cell would return from the inferior superior vena cava, the volume would be of course increased. When the volume would be increased, so increased volume of the blood is coming into the heart as compared to the volume that was of course leaving the heart. So this would of course cause the increase in the cardiac output. This increase in the cardiac output would of course increase the renal pressure, the atrial pressure, which will of course continue, which will of course cause increase in the renal excretion, and this situation will persist unless or until this particular total peripheral resistance is reduced. So in case when this particular peripheral resistance is reduced, the atrial pressure will decrease, the extracellular volume will decrease, the blood volume ultimately will be decreased as well. This would of course cause a reduction in the venous return, and the cardiac output would be decreased. This decrease in the cardiac output would of course normalize the atrial pressure as well, and the renal pressure would of course will turn to its normal excretion rate. So the renin-angiotensin system. The renin-angiotensin system plays a very important role in the regulation of the sodium, potassium, and the water reabsorption. So what happens is that the liver would, of course, release the angiotensin and the angiotensinogen. This will move and will be converted into the angiotensin. This angiotensin will move and will be converted into angiotensin 2 by the adrenals. It would cause the stimulation of the aldosterone. Now, aldosterone will have an effect on the heart, arteries, and the kidneys, but aldosterones will be in the kidneys. In case of kidneys, they will be causing the retention of the sodium in the water. So, the aldosterone is being secreted when there is a low amount of the sodium, or they will be secreted when the water has to be reabsorbed, or they will be secreted in the case of excessive quantities of the sodium or of the potassium. So the sodium and the water retention will cause the increased blood pressure, and increased blood pressure will be caused because of the vasoconstriction, because whenever you decrease the diameters of the arteries, diameter of the arteries, it would cause increase in the blood pressure, of course. So this figures also explains the renin-angiotensin system. It explains that when the renin, of course, is released from the kidney, it is being converted by the liver. When the angiotensinogen is being released, it is converted into angiotensin 1. And this angiotensin 1 is converted into angiotensin 2, and these are the enzymes that are involved, which will, of course, be causing the conversion from its precursor to the next particular molecule. Now, angiotensin 2 plays very important functions by acting on the adrenal cortex, causing the release of the aldosterone. These aldosterone, along with the angiotensin 2, would of course cause the vasoconstriction. This means that the diameter of the volume, the diameter would of course reduce. This would increase the blood pressure. Aldosterone, on the other hand, will move towards the kidney, and it would cause the sodium and the water retention, which would of course cause the increase in the PV. Now, what happens is that when there is decreased blood flow or decreased sodium ions, the juxtamedullary apparatus of the kidney will secrete the renin, which will of course convert angiotensinogen into the angiotensin 1, which will of course be converted into angiotensin 2 that we have discussed in the previous slides as well. Now, angiotensin 2, as we have discussed, is a powerful vasoconstrictor. It will cause the vasoconstriction to increase the blood pressure. Now, this would of course move into the adrenal cortex, where the corticosterone will be released and in the form of aldosterone. Now, the aldosterone is basically the main regulator of the sodium retention. It is also important in the regulation of the water reabsorption, and along with that, the potassium excretion. So in this lecture, we discussed the regulation of the excretion of potassium along with its extracellular fluid concentration. We discussed an overview of what happens in case of the renal potassium excretion and under what circumstances potassium excretion would be increased. We discussed the potassium secretion by the principal cells of the late distal and the cortical collecting tubules, and in that we discussed under what circumstances the potassium will be moving from renal interstitium into the principal cells through the potassium ATPase, sodium potassium ATP pump, and after it has moved inside, the rise in the concentration would establish an electrochemical gradient or the concentration gradient, which would of course cause the diffusion or the leakage of the potassium from inside the principal cells through the basolateral membrane into the tubular lumen. Now, this would of course be occurring as a result of increased potassium intake. We discussed the factors that regulate the potassium secretion and under what circumstances, for example, aldosterone, under what circumstances they will be of course increasing the potassium concentration. We discussed what happens, what changes occurs in case of the metabolic acidosis and alkalosis, and we discussed it for both potassium and calcium, along with this discussion with the magnesium ions. We discussed after that how the calcium ion concentration is being controlled and regulated. We discussed the regulation of the magnesium ion concentrations and how it is being controlled and under what circumstances they are being released. We'll discuss the primary parts, are the portions at which the magnesium ion concentration is being reabsorbed and their preferences. Let's say we discuss that the from the proximal tubules, its reabsorption is about 25%. From the loop of Henle or descending loop of Henle, its reabsorption is about 65%, which is basically opposite of the concentration reabsorption of the both sodium and potassium. We discussed the pressure natriuresis and the pressure diuresis, which we discussed how the pressure natriuresis leads to the pressure diuresis, that is increased sodium excretion would be of course causing the increased water excretion as well, and we discussed the renal body fluid feedback mechanism, and last but not least we discussed the renin-angiotensin system, and we discussed how it plays a very important functions in the natriuresis and in the establishment of the blood pressure or in the vasoconstriction as well, and we finally discuss how this renin-angiotensin system plays an important role in the secretion of the aldosterone, which along with the sodium and the water retention causes the secretion of potassium. Now that is all for this particular lecture. For more lectures, keep watching sqadia.com. Thank you.